This paper looks at COVID infection in the oral cavity and in saliva. This was a mixed autopsy and saliva sampling study for COVID infected individuals, where they found that in asymptomatic individuals, they had saliva that was able to transmit COVID, and that the higher the COVID presence in the saliva, the more likely you were to have taste loss symptoms. Where ultimately they showed that the oral cavity is an important site for COVID infection and that saliva is a potential route for COVID transmission. So, again, COVID infection was confirmed using autopsy and outpatient samples, where saliva from asymptomatic individuals with COVID demonstrated the potential for viral transmission, where the mucosa of the oral cavity and oropharynx are likely gateways for viral replication and infection. Interestingly, there wasn't one singular target in the mouth that COVID focused on, which suggests that multiple oral epithelial cell subtypes are susceptible to infection, or in other words, that your COVID infection in the mouth can happen in multiple places. This includes places like the buccal mucosa, which is this area of the cheek, the ventral tongue, which is the underside of that tongue, the dorsal tongue, which is the top surface of your tongue, the soft palate, which is the roof of your mouth, the palatine tonsil lining mucosa, which are those big bulbs in the back. That's not the uvula, but the things next to it. The linguinal tonsil lining mucosa, which is that area with the giant bumps on the back of your tongue, and the linguinal tonsil crypt. Now, interestingly, all of these different parts of the mouth have high levels of ACE2 and TMPRSS2 receptors. These are two of the main receptors that COVID binds to, which again means that all of those locations are potential binding sites for COVID infection in the mouth. Additionally, during histology, they found that COVID, which is denoted by the red in these images, is highly present in both the minor salivary gland and in the parotid salivary glands, which means that COVID can infect and replicate in the salivary glands and the mucosa, where COVID was detected in 57% of the salivary glands tested, with a trend towards higher viral loads in the minor salivary glands. These results establish that the minor and major salivary glands are susceptible sites for infection, replication, and local immune cell activation, where these findings raise new questions about COVID pathogenesis, including one, whether this is primarily a nasal first infection that spreads to the oral cavity, and two, the possibility of an oral first infection via droplet or aerosol inoculation or fomite ingestion, and three, whether the pattern of infection affects disease severity and host immunological responses.